Hi everyone, this is Peter Dorsky in RAF 6204. This is my end of phase 2 briefing package presentation for PD-INS, insulin cellular rDNA origin, 100 units per milliliter solution for subcutaneous injection. Note that cellular is a completely fictitious name, although my study designs were based off of a couple other rDNA origin. So the contents of this presentation are an introduction, general information on the product, purpose and objectives, list of attendees, phase 1 and phase 2 summary, and the bulk of it will be focused on the proposed phase 3 program and the phase 3 study design and justification for some of my design components. So a very brief introduction to diabetes. In 2012, 29.1 million people in the US, or 9.3% of the population, had diabetes. 2010, diabetes was the 7th leading cause of death, and approximately 1.4 million Americans are diagnosed annually with diabetes. Treatments for diabetes include diet, exercise, and therapeutic prescription drugs or biologics, which typically include either prescription pills or insulin. Over 50% of diabetes patients are currently on some sort of medication, with a little over 50% taking pills only and 17.8% taking insulin only, 13% taking both insulin and pills. Some general information on insulin, and note that I'm not a biochemist, so this is the super condensed version of how insulin works. Basically, it's a peptide hormone produced by the pancreas. It's the endogenous ligand for the insulin receptor, which is present on the surface of cells which respond to insulin. And the activation of the receptor is triggered by the binding to the ligand, which leads to autophosphorylation, initiating the signal transduction cascade. And the most important and clinically relevant effect of that is that it causes the uptake of glucose into the cells, which lowers blood sugars or blood glucose levels. Onto the name, presentation, and indication of PD-INS. Recombinant insulin is technically a biologic, but historically it's been regulated as a drug and approved as an NDA under Section 505 as opposed to a BLA under the Public Health Service Act. It is a pre-filled pen integrated with 3 ml cartridges made of USP Type 1 glass. Each pre-filled pen is intended for use by a single user, and you can see that it's disposable, multiple, and variable dose. Designed with commercially available insulin pen needles, which are also disposable. The indication is to improve glycemic control in adults with diabetes as either monotherapy or concomitantly with oral antidiabetics or fast-acting insulin, or both. And I have here an image of the Eli Lilly QuickPen, which is has a pretty common design to most insulin pens. And after removing the cap, the rubber seal is exposed, and you can see the needle parts here and how it's assembled. And basically, you have your dose knob. Twist your dose knob to select your dose of insulin. Attach your needle. Stick yourself. Depress the knob. Hold it for 6 seconds or whatever the instructions for use tells you to hold. Then hold for another 6 seconds while the dose is administered, and that's pretty much how you deliver a dose of insulin. The purpose and objectives of the end of phase 2 meeting with the agency is to reach agreement with FDA on the following: pivotal study designs and safety and efficacy endpoints for phase 3 studies, the proposed device development plan, the proposed request for deferral of a thorough QT study. And these last 4 I just kind of added because they would something probably worth discussion with the agency at an end of phase 2 meeting. Proposed plan to request deferral of pediatric studies under PREA. Proposed comparability assessment between the formulation used in clinical trials and commercial formulation. Proposed establishing shelf life based on drug product stability data obtained from the development and clinical lots using the same process as that of proposed commercial formulation. The list of attendees to the end of phase 2 meeting from my company, PD Drugs, which is making a biologic. I just noticed that now. And the fictitious names can be ignored, but the positions, the head of global clinical research, senior director R&D, head of Reg Sci, biometrics director, senior director of complex products, devices, global RA and operations, but basically people who would be able to make a decision at the meeting. So, and also subject matter experts who can have discussions with the agency, and having these people there will help to come to an agreement with the agency as well as help make commitments, if commitments were to be made based on either study design or possible post-approval commitments. I also have a list of requested FDA attendees, and the only one I mentioned by name is the DMEP, the Division of Metabolism and Endocrinology Products Division Director, Dr. Jean-Marc Gutierrez. The other ones are just placeholder names, representatives that I would like to attend, such as the RPM, representatives from the Office of Regulatory Policy, Manufacturing Control Team Leader, CMC Team Leader, Lead Medical Officer from DMAP, Clinical Pharmacology Reviewer, Biostats Reviewer, and a representative from CDRH to respond to my device questions. And I actively chose to not ask for a representative from the Office of Combination Products, even though a prefilled pen is technically a combination product, and the reason for that is because there is enough guidance and regulatory precedence that I don't think we need anyone from the Office of Combination Products to give their input on how a prefilled pen should be developed. Moving quickly through the Phase 1 and Phase 2 study summary, here's a list of 14 studies that were part of my early clinical development program. Key messages from the Phase 2 PK/PD studies specifically in patients are that the glucose lowering effect of PDINS was proportional or linear to dose. PDNs and IGLAR, insulin glargine, which is a long-acting insulin and a good comparator for PDNs, had PD profiles which extended beyond 24 hours, and PDNs had a glucose lowering effect duration of greater than 42 hours, and that 42 hours would actually make for an interesting clinical study that I didn't take advantage of, but that Tresiba, one of the clinical programs that I based mine off of, did, and that is to actually aim for a regimen of 3 times a week as opposed to daily, with a 42-hour duration of exposure time. You actually might be able to get away with dosing 3 times a week as opposed to daily. But the fourth bullet point is the PK profile of PDNs is flat and stable compared to that of insulin glargine, and the safety profile of PDNs is similar to that of IGLAR. And that last one's kind of a bold statement, but that's obviously something you would test for in your confirmatory Phase 3 trials. Now on to the Phase 3 proposal. It consists of 7 randomized studies, 2 studies in patients with type 1 diabetes, 5 studies in patients with type 2 diabetes. And they do assess different things, which allows me for a rather broad indication in a large group of patients. Notice that for each indication, type 1 and type 2, I do have one cardiovascular outcomes trial to assess major adverse cardiovascular events. But the study I have underlined here is the study we're going to be going through, and that trial will assess the safety and efficacy of PDNs in patients currently treated with once daily insulin glargine and either oral medications or insulin aspart or both. So the title for that underlined study is a 52-week randomized. Purpose and objectives. The purpose of this trial is to compare PDNs with insulin glargine plus insulin aspart. Insulin aspart is a fast-acting insulin typically taken immediately before you eat, whereas insulin glargine and PDNs, insulin Soleag, are long-acting. They're also taken before you eat, but they have exposure or peak PK times of usually longer, 12 hours, 24 hours. With or without metformin and pioglitazone, which are oral anti-diabetics, so the subjects may or may not be taking oral anti-diabetics. That's part 1, 26 weeks, followed by another 26 weeks safety extension, investigating the safety of PDNs compared to insulin glargine. There will be 1,000 subjects in this study, patients with TDUTM. Okay, this is patients currently taking once daily insulin glargine as monotherapy in combination or with insulin aspart, or oral anti-diabetic, or both. So my study design does have a 6 to 8 week run-in period for patients who meet screening criteria, and during this run-in period, patients will be educated on diabetes and optimized for compliance with exercise and their current treatment. This will allow for what the guidance calls a stabilization of parameters of metabolic control, so that the magnitude of the effect of different doses of the product can be most accurately estimated. Because reinforcement of compliance with the treatment regimen is enforced so strongly during the trial, the absence of this run could lead to bias and suggest efficacy related to the treatment during the trial is not representative of the real-world setting. The study is designed as a treat to target, with treatment to be delivered reaching a fasting plasma glucose of less than 90 milligram per ml. Here's my inclusion criteria for this trial. I think it's pretty typical for long-acting insulin in adults. I did highlight number 4 because those currently receiving treatment for diabetes and on a stable dose for greater than 3 months, there's still going to be a run-in period for those, but I think this will help to reduce the heterogeneity of my study population and give me more accurate results. And my exclusion criteria, I won't go through each one individually, but I will focus on a couple of them. Number 3, the cardiovascular risk. In the last 6 months, those individuals will be excluded from the trial. People currently on oral antidiabetics such as TZD, rosiglitazone, and DPP-4 inhibitors. And the reason I am excluding these is because those drugs have a known effect on the glucose profile, the HbA1c and HbA1c assay results. So I want to exclude them once again to reduce heterogeneity of my results. And patients with severe hypoglycemia, defined as one severe hypoglycemic event within the past 12 months. And the definition comes from FDA guidance, an event requiring assistance of another person to actively administer carbohydrate, glucagon, or other resuscitative action. And more exclusion criteria, nothing here worth noting. And withdrawal criteria for patients already randomized into the trial. Moving to the underlined ones, the patient's pregnant or becomes pregnant, that they become high risk and treatment is discontinued. For hypoglycemia or hypoglycemic events, which in the opinion of the investigator pose a safety concern, and lack of treatment effect at 12 weeks as determined by the protocol. The efficacy portion of the study is 26 weeks long, but at 12 weeks there will be an HbA1c assay and an interim analysis. And so if patients are treatment shown to be ineffective at 12 weeks, they would be withdrawn from the study. To the study procedures, all test articles will. Lastly, we have the endpoints, and for part 1, our primary endpoint is the change in baseline HbA1c at week 26. And I have here a note that HbA1c is the efficacy endpoint of choice for determining efficacy in type 2 and as well as type 1 diabetes patients, and that's from FDA guidance. Your key secondary endpoint is going to be your immunogenicity testing, and that endpoint is incidence and change from baseline in the relative levels of anti-drug antibodies, and the incidence and change from baseline in the relative levels of anti-HCPs. Also, the safety endpoints will be the rate of treatment emergent adverse events and the mean of 9-point SMPG profile at 26 weeks, the rate of hypoglycemic events per 30 days and occurrence of hypoglycemia, and the change in 8-point SMPG profile, as well as the device-related safety assessment. And that's a user safety assessment. And our endpoints for part 2, primary endpoints, are the rate of hypoglycemic events and the rate of TEAEs. Our secondary endpoints are change in week 26 glycosylated hemoglobin, so you're still your HbA1c, still going to test for efficacy in the second part, and the mean of 9-point SMPG profile 52 weeks and change in 8-point SMPG. And SMPG is self-monitored plasma glucose and self-monitored blood glucose, so those are patient monitored, typically in a diary, so I'll measure those as well. So I'm looking forward to your questions, comments, concerns, and criticisms, and thank you for listening. looking forward to seeing all of you on the discussion boards.